Dogs. Can I say thrombospondin and matricellular? I'm Joanne Murphy-Ullrich. I'm a Professor Emerita at UAB School of Medicine, Department of Pathology. I've served on the Executive Council of ASMB for many years. I was on the first council, and I'm currently Editor-in-Chief of Matrix Biology, Matrix Biology Plus. So this is my scientific home. Do I have a hobby? I have a dog and a grandson.
So in addition to having good grandma time and taking long walks with my dog Nora, I've gotten into growing tomatoes over the last few years. One particular variety that I like to grow is something called sun sugar. It's a small cherry tomato, develops fruit early in the summer and very abundant. In fact, I had almost a full plant of fruit when we had our first frost in November, and I was able to harvest the fruit and kept it in the kitchen window, and it eventually ripened, and I had my last tomato, homegrown tomato, on Christmas Eve. Pretty sweet.
Thrombospondin 1. Yes, our very serendipitous discovery that thrombospondin binds to TGF-β, and then the more interesting aspect of it that not only does they bind, but that TSP1 binding to latent TGF-β converts it to the biologically active form of the molecule. And that's really been the primary focus of my career for 30-some years. Cell culture, Western blot, ELISA, yeah, very standard, low-tech things. Nothing, astounding like everybody's doing now, but histopathology, immunohistochemistry. Oh, this is very interesting. So as a grad student, I was in a lab, and I thought I wanted to be in immunology, and this is in the late 70s. And people didn't have a way of talking about what immune cells were between labs. They just defined them by antibodies. It was before, cells were cloned and had markers. And one day, an assistant professor associated with my professor came in the lab, and he was carrying the New York Academy of Science report from this meeting on fibronectin. Dean Mosier gave it to Terry Oberle, who was my Ph.D. mentor. And Terry said, hey, have a look at this. And that was it. So my Ph.D. thesis was actually on autoimmune response to fibronectin and laminin. And then I stayed in Madison. My husband was still working on his Ph.D. in English, and went to work with Dean Mosier, and Dean said, I'm beginning to work on this new protein, thrombospondin 1, and that was it. Yeah, Terry D. Oberle was a renal pathologist. He was my Ph.D. advisor. Dean Mosier was my postdoc advisor, and Dean was also on my thesis committee, because we didn't have a Western blotting apparatus in our lab, so I had to go across the street to Dean's lab to do my Western blots. And that's been a lifelong, wonderful mentor relationship. And then when I was in Dean's lab, I was looking at thrombospondin 1 binding to endothelial cells, and we thought that naively, the thrombospondin receptor, not realizing there's multiple receptors, we thought it was a heparan sulfate proteoglycan. So then I moved to UAB. At the time, UAB was just this wonderful nidus of matrix research. There were collagen people, there were lots of proteoglycan people, and some MMP scientist as well. So this was a logical place for me to go, and I worked with Magnus Hirk for several years, and Magnus gave me the opportunity to bring thrombospondin to Birmingham, do my work there. That's where I made the discovery that it activates latent TGF-β. I also worked with Ann Woods. She was studying syndicans and focal adhesions, and we showed that thrombospondin causes focal adhesion disassembly. So I had kind of those two avenues of research going. And a tenure track faculty position opened up in the early 90s in pathology at UAB, and it just worked out. Been there, I think a total of 37 years before I retired officially. Dean, yeah. He's just a superb protein chemist, very rigorous, just knows everything. I think he has photographic memory, so he would know more about what you did in your experiment than you would. So follow your passion, because this is a difficult field, lots of ups and downs. Believe in yourself, because you're going to have a lot of people say no, but intrinsically you have to carry within you that the work you're doing is meaningful, and just kind of be stubborn, keep at it. Not to a fatal extent, but just believe in your work in the face of negative comments, and yeah. It'd be hard to distill it down to one thing. I mean, it's there's so many good friends, and that have been friends for decades now. There's probably some good stories, but I wouldn't want to be publicizing that. But it's nice to come to this meeting, see the direction that things are going, the enthusiasm among the trainees. So it makes me feel good about the direction of the science and the matrix field. Because we're always a little niche group, and we struggle to get the word out. But it's good to see that there's a new generation carrying on the work.